And as we kick off this session, I'd like to remind you that as you have done in Morton's session, please ask questions to the speakers in the Q&A tab as you listen to the presentations, and we will be saving all questions to the discussion and Q&A part of the session. And to chair this session, we have Adam Baker, Director of Science at Christian Hansen, joining me on stage. And Adam is responsible for the research of current and next generation probiotics, as well as building up a strong portfolio of scientific data to support the commercial probiotic business. Adam focuses on the role of bacteria throughout life and the impact that they have on our health. So welcome, Adam.
Thank you so much, Sarah. And I'd like to thank you all, thank the organizers for this great event. It's a real privilege to be here today.
Yep.
And just to hear Morten talk and to actually then share some of the exciting science which is going on in the Medicon Valley region. So I'm really pleased to be chairing the first session.
Yep.
And I think I should now say that we actually have a poll.
Exactly.
Do you want to say how it
Sure,
works?
yes. So for you who are on Hopin right now, there is a poll that is launched on the right side of your screen in the Stage tab under Polls. And Adam has created a question in the poll for us today. To what extent has the current global pandemic made you more aware of or even want to actively improve your immune health? And there are a couple of options to choose from. Feel free to respond, and I'll hand the session over to you, Adam.
Fantastic, thank you. And I'll get to see actually how the results come out.
Absolutely, yes, they will update.
Okay, it will update, that's great. Okay, well, thank you, everybody. I'm so pleased to be here, as I've said today, to chair this first session and really to actually have the opportunity to sort of highlight and introduce some great research that's going on within the region. The title of the session is really sort of Immune Health and the Microbiome in the New Normal. And I want to touch a little bit on this. I think Morten's keynote address was fantastic and touched on some of the points that we want to take forward. And really, I have a few slides to take. And Morten brought this up, the microbiome, how important is it? Well, I think it is absolutely key. We're going to be hearing about this throughout the day. And we really are trying to share and actually sort of take in now how we actually think about the microbiome and that we're living in a sort of microbiome universe. Morten's touched on it. I think living in a symbiosis, the microbiome ecosystem, is in us and on us. Now, there's a lot of focus on the gastro microbiome. We have other microbiomes as well that we'll be talking about today. The oral microbiome. We're thinking about microbiomes in agriculture, in plants. These are all very, very key. But I think you heard some of the science that Morten's pushing forward, how we can really start to think about what we do in terms of microbiome. Think about therapies. We think about prophylaxis. Now, I work at Christian Hansen, where we're very, very proud of our natural solutions, how we work with food. And I think Morten called it sort of low-tech. But I think the food and the nutrition can be so key and important. We were touching on companies and thinking about how we can start to look at the microbiome, study it, diagnostics. We're hearing about small companies that actually focus on these things. But I think what we want to do is we want to actually see and we want to actually understand how the microbiome is working, it's within us and on us throughout life. And in this simple example I'm showing you here, we see, even when we're adults, the sort of mismanagement sometimes that goes on, day-to-day factors, stress, lifestyle, these all affect things. And what we're trying to do, and that's what Morten was actually pushing forward with some of the companies he works with, always trying to get dysbiosis away and sort of bring microbiomes back to a sort of normal and diverse state. So these are the things we actually think about. But some of the sessions and some of the research I get very privileged to be actually highlighting today will actually be focusing on sort of a little bit how we think about the microbiome and the development of the microbiome from birth and how important it is for certain aspects. And I think certain aspects, particularly when we think from birth, the microbiome and how we evolve with it and our immune functions. Our immune system is evolving in collaboration with the microbiome. They're intimately linked. And we have to think about this very carefully. And I'm very happy today that we're going to have really some speakers that actually I'm very proud of from the Medicon Valley region, talking about different aspects of how the microbiome affects and works with the immune system. Your neurological systems are also developing at the same time with the microbiome. So this development and actually what is going on early in life and from birth is critical and key and can actually lead to diseases or symptoms and syndromes that we actually touch on, that you actually take through life. So how important it is to actually understand the development of the microbiome and other systems. And then later in life, as we're adults, what type of effects, what type of factors are actually affecting these. So these are all key things. We're going to touch on these and we're going to be looking towards it. So I think that that's so important. I'm just going to look now and see if anyone's actually answering the poll and where do I actually see the results. Sarah, if you show me here. So
Yes, and I think we have actually close to 100 answering the poll, and I'm just going to activate this. There we go.
Okay, very good. Great. And actually what I'd like to see is actually, as we're understanding immune health and actually what are people actually saying as we're going in live, that actually to a very large extent or a large extent is actually what people are actually thinking. And I'm hoping that actually through the talks you're going to hear, you'll actually even be more believing in how important microbiome and immune health are. But that's really interesting to see that actually really we already are very aware of where we are. So before we get to the speakers, because I'm going to be very soon introducing the three speakers that I'm very pleased to announce, I just wanted to sort of provoke again. We are in a new normal, immune health and the microbiome. I think this is someone that most people actually recognize on screen. Has been very involved in the recent pandemic. But I think maybe not so many people are aware of it. Anthony Fauci is actually publishing, and even last November, while he had many other publications and things he was actually busy with, was actually publishing on how we really need to work with the microbiome and actually rethinking about how he writes a review on thinking about immune-related diseases and the role of the microbiome. I think this is key. People are very aware of these things now. And actually, from the scientific point of view, I think Morten could be touching on it in some of his companies as well, how we think about vaccinology as well, what is actually happening, how people are responding to vaccines. All of these things people are becoming intimately aware of now as we are trying to live in these new times. And the importance of the microbiome, how you actually affect and what actually goes on is so key. So with that, I really need to start to sort of introduce the three speakers that we have today. And I work at Christian Hansen here in Copenhagen, but one of the things that Morten actually touched on is the actual access and the great research that's going on around the area. So we have three speakers today I'm very proud to be introducing. We have Professor Asa Hawkinson, who's going to be talking and touching on sort of how the microbiome and the immune system are working together, really getting into the details and allowing us to understand in a much closer way what is actually happening. We also have Professor Daniel Agard, who will be talking to us. He will be the final speaker in the session, actually talking to us a little bit about the sort of using clinical sets, one of the advantages we have in the Copenhagen area, clinical trials and clinical cohorts, and understanding types of autoimmune related diseases. And Jakob Stockholm, who will be joining me on stage soon, who will actually be touching on, again, the use of clinical cohorts in the Copenhagen area and what we're actually thinking about and how these can actually address immune-related disorders, asthma from actually childhood. Again, linking this back to the microbiome. So I think as we sort of finish, or what I would like to say is that there is so much work going on. There's so much emphasis at the moment around the sort of microbiome and understanding. You've heard it from Morten. You're going to hear it through the day as we touch on these different things, as we think about it. What I would like to say is, of course, we can, and we're understanding the point of view of how we can actively take a role through food, through nutrition, through medications, through diagnostics that Morten also touched on. There's some fantastic young companies thinking about diagnostics to understand the microbiome. It's key. It's an incredibly exciting time to be studying the microbiome. It's an incredibly exciting time, an important time to understand how the microbiome is influencing, particularly, and I'll be touching this session, the immune health, but all other areas of sort of health as we go on. The importance from birth and childhood and how that can actually influence your long-term health factors. How antibiotics are causing maybe some situations, some problems that we're actually now living and dealing with, and how we can actually address those through either nutrition, through medications, through sort of disease treatments. So I think with that, I would like to now actually, I think the poll is finished, and yes, everyone is actually saying that to a large extent the immune health is actually linked to microbiome, and that is actually great, and that actually people would actually think about actually trying to actually improve their immune health. So with that, I think now I would like to actually hand over. We're going to be going first to Professor Asa Haukinson, who is going to introduce us to her work and the presentation. So with that, over to Asa.
Thank you very much for the presentation and for the kind invitation to speak at the Microbiome Summit 2021. The human gut microbiota is considered a major modulator of the immune system during development and in health and disease, and in return, the host immune system has evolved mechanisms to maintain its symbiotic relationship with the microbiota. The ability of microbes to induce an immunological reaction, both locally and systemically, requires specific sensing of the microbes and complex feedback loops between innate and adaptive components and cells of the immune system. A very simplified description is that the response is initiated in the lumen through an interaction between the antigens and the epithelium, and through capturing of the antigens by M cells or dendritic cells from the lumen side to lamina propria. The left figure illustrates some examples of how the microbiota influence the innate immune response, primarily through cellular structures, such as LPS on gram-negative bacteria, and metabolic products, such as short-chain fatty acids. Recognition by intestinal epithelial cells induce, for example, secretion of antimicrobial peptides and cytokines, and the recognition of flagellin and sphingolipids by dendritic cells can lead to activation or inhibition of lymphoid cells. The right figure shows examples of microbial influence on the adaptive immune responses. Signals from commensal bacteria induce interactions between epithelial cells, intraepithelial lymphocytes, antigen-presenting cells, and lymphocytes located in lamina propria and also in organized lymphoid tissues, such as follicles, Peyer's patches, and mesenteric lymph nodes. The interaction will lead to activation and differentiation of effector cells, such as IgA-producing plasma cells, cytokine-producing T helper cells, and regulatory T cells. All reactions are induced by complex signaling pathways. The human gut contains a highly diverse microbial core community. Despite the resistance of the microbiota to colonization by ingested organisms, many foodborne bacteria in fermented food, in probiotic products, but also in other consumable food products, are capable of transient integration where they may impact the composition and activity of the resident microbiota. Foodborne bacteria can therefore be regarded as part of our transient microbiome that is both highly dynamic and individualized, and its composition is the result of recent exposure, the condition of the host gut ecosystem, diet, and the properties of the ingested organisms. Even though stimulation of the immune system responses by probiotics have been reviewed to a large extent, further research are needed. The possibilities of foodborne bacteria to induce immune responses are, however, still unknown, even though the mechanisms would be the same as for commensals, which is illustrated in the right part of the figure, where foodborne bacteria are transported and presented by M cells and dendritic cells respectively, leading to activation of lymphocytes. I would now like to briefly present some of our studies. Ready-to-eat leafy green vegetables are popular products, but also one of those products providing a high bacterial intake at consumption. In this study, which is not yet published, human monocyte-derived dendritic cells were stimulated by E. coli strains and by native plant commensals, all isolated from commercialized products that we normally consume. And activation markers CD80, CD86, and HLA-DR were investigated. The aim was to predict a possible pro-inflammatory effect of these bacteria in humans. After stimulation, a relative increase of all marker levels were found, indicating an activation in response to the E. coli strains, but also in response to two of the commensal strains belonging to different genera called TP2 and TR in the figure, which could be consistent with the pro-inflammatory response. If the plant commensals do induce an activation, also in vivo needs further evaluation. Microbial colonization during infancy plays an important role in development of the immune system, as they are established in parallel. An important factor in the development is early life nutrition. In the present study, we analyzed endotoxin concentrations and quantities of viable bacteria and porridges and milk cereal drinks. We found considerable amounts of endotoxins ranging from 200 EU per milliliter prepared MCD to 7,300 EU per gram prepared porridge. And we also detected viable cells belonging to the dominating families presented in the right figure. Live bacteria belonging to the family Bacillose were found in all our products, sometimes generating intake of high doses after consumption of 1 portion or more, which is a common consumption quantity for children in Sweden. For both members of the family Bacillose and for endotoxins, previous studies are indicating negative impacts on the intestinal barrier functions and immune modulating properties. The results reveal the need for larger studies on the prevalence as well as further studies of the physiological effects after long-term consumption by children to evaluate if the microbiota and the intestinal permeability are actually affected. Now we proceed from children to elderly, and in the present study we analyzed the effect of a short-term nutritional deficiency on microbiota and immune system interactions. The aging processes severely cause a decline in the functionality of the immune system, a changed gastrointestinal physiology and function, which in turn affect dietary intake, and also a modified microbiota composition, distinct from what we find in younger individuals. The changes increase the risk of malnutrition and immunosenescence, but how fast the response is induced is not known. After 4 weeks of poor nutrient intake, we found significantly affected population sizes of activated regulatory T cells, of macrophages and dendritic cells expressing TLR2 and TLR4, and on lymphocytes expressing homing to the gut. The populations were also found to correlate with certain bacterial groups in both Peyer's patches and mesenteric lymph nodes, as you can see in the table and in the PLS plots. Increased expression of TLRs may lead to increased recognition and enhanced or changed responses due to an abnormal bacterial composition. The results from the study indicates that only a short-term nutritional deficiency is needed to induce an immunological response and a modified microbiota composition. Epidemiological studies have found a correlation between red and processed meat consumption and an increased risk of colorectal cancer. And in these cases, there is also a link to inflammation and dysbiosis. In this study, 5 plant antioxidant preparations were added to meatballs, and pro-inflammatory markers and microbiota composition were studied after 4 months in a model of chronic inflammation. We found that the bacterial diversity index was affected in both colon and in the small intestine, and when analyzing the terminal restriction fragments, differences were found after meat consumption. The microbiota composition was also further changed after addition of antioxidant preparations. Differences in cell populations of both Peyer's patches and mesenteric lymph nodes were detected, and correlations between certain bacterial groups, infiltrations of neutrophils into the intestinal tissue, and leukocyte populations were found. The correlations found were more pronounced for regulatory T cells, macrophages, and activated T helper cells. The screening was conducted at an early stage of induced chronic inflammation together with processed meat consumption, but finding initial differences in gut microbiota and immunological responses could be of valuable interest for further studies during longer periods of time. In another model of induced acute colitis, the population of regulatory T cells co-expressing CTLA4 was increased in the colitis group compared to the normal group in both Peyer's patches, spleen, and mesenteric lymph nodes. In the study, no significant difference in bacterial diversity was detected, but changes in occurrence of certain bacterial groups, as well as increased amounts of total bacteria, Akkermansia, and Enterobacteriaceae species, but decreased amount of Lactobacillus species were found on colonic mucosa. As for previous studies, immunological responses correlated with findings from the microbiota analysis also in this evaluation. Lastly, I would just like to mention the immunological response in children with celiac disease autoimmunity after probiotic consumption. I will not go into any details since Daniel Agard will present this study further. After oral administration of the probiotics, significant changes in lymphocyte subsets were observed between the treatment and placebo groups and within the placebo group over time. Interestingly, the difference of most lymphocyte subsets found in the placebo group was similar to what is found in patients with active celiac disease, indicating a progression of disease development that we could not observe in the probiotic group. In the study, several different phenotypes were affected on population size. For example, the populations of regulatory cells identified as CD4, CD25, and FOXP3 positive cells, and CD4, CD25 high, CD45 low, and CCR4 positive cells increased in the placebo group, but remained unchanged in the probiotic group. The findings may be explained by a down-regulatory effect of the two lactobacillus strains used as probiotics in this study, and the increase found in the placebo group for these populations could be an attempt to dampen the ongoing inflammation in the children. Another finding of importance was the peripheral changes in NKT cells over time in the probiotic group, which was not observed in the placebo group, but also population of cytotoxic T cells or CD8 positive cells and T helper cells, which are CD4 positive cells expressing memory functions, were affected. Altogether, the results indicate that oral administration with the probiotic strains may modulate the peripheral immune response in children with celiac disease autoimmunity, but further studies are needed. Before ending my presentation, I would like to thank the collaborators from academia and industrial partners, and thank you all for your attention.
Great, and thank you, Asa, for the great presentation. Just to remind everybody that we'll be doing the questions in the Q&A at the end of all three talks, so you can please write, and I can already see we have questions coming in, but we will take them all at the end of the three talks. And now it's my great pleasure to introduce Jakob. And Jakob, would you like to take the stage and introduce your talk, please?
Thank you very much, Adam. Yeah, my name is Jakob Stockholm, and I'll be talking here today about the early life microbiome and what that means for later disease development, namely asthma and allergy. And I'll present some results for you here from our COPSAC birth cohort. So that is for Copenhagen Prospective Studies on Asthma in Childhood, a big collaboration between Danish Pediatric Asthma Center, Næstved Hospital, but also collaborators from the universities, Copenhagen University and DTU. Today I'll be framing about what happens with the microbiome very early and associations, I must admit, to disease. So why do research in childhood asthma? Childhood asthma is the most common chronic disorder in childhood. It's the most common cause of hospitalization in children, medication use, and also contact to the physician. And it occurs as these attacks of breathlessness, wheezing, and coughing. And to show how often this occurs, so 20% of children will have asthma before school age. It will some will grow out of it, and about 7% after school age. The same kind of trend is seen for the childhood eczema, and allergy is the opposite, where it's very infrequent in early life, but then occurs much more during later life. And these diseases are something that you can actually study because it's in quite high prevalence and all kind of immune-related disorders. So very interesting to study in early life. So with these microbial exposures and later disease, there's been numerous studies performed by us and others on different environmental factors that might actually influence the child by microbial exposures. Factors such as living in close contact with other children in a daycare setting, having our good friends, the household pets in the home, then growing up on a farm seems to be protective. The same accounts for growing up in a very bad sanitary condition. Being born by cesarean section is a risk factor for disease, and the same accounts for antibiotics use in early life. And our question is, of course, can we take all these factors and try to see whether they are to some extent mediated through microbial changes very early on? So for these diseases to develop, you need to have some sort of heritability, but only if you're hit by, you can say, wrong exposure or maybe a right exposure, the disease will not develop, even though you're susceptible to the disease. And here the microbiome comes in as a very interesting target, because we know that the microbiome, as we also heard in the other talks, are interacting with the host. It can modify the local environment, perturb the host defenses, interact with the whole immune system of the child, which is developing at this time, and also, of course, produce a lot of bioactive substances. So where do the bacteria that colonize the young child originate from? So when you're born, you're exposed to mother, gut, and vaginal. It will colonize the upper and lower airways. You'll be colonized in the gut. You'll be colonized on the skin. And furthermore, you're also exposed to a variety of bugs through the home environment, the dust, wherever you live, there are bacteria and fungi. So in COPSAC, which is what I'm going to show you here, this is our COPSAC 2010 cohort, 700 children, follow extremely close, 16 planned visits in the first 10 years of life. They come to us every time they're ill, so every diagnosis of asthma and allergy and eczema are put on by the research physicians. We measure all exposures we can in early life. We do pregnancy interventions, and then we follow the children, which are all healthy in the beginning, and see who develops asthma, who will develop allergy, eczema, but also looking at factors like obesity, neurodevelopment, psychiatric disorders, and so on. And we try to take a lot of samples during early life, so we can try to see what may go wrong or what may go right, and try to profile them for genomics, immune profiling. Then we have the microbiome, the virome, epigenomics, metabolomics, and so on. And that is trying to put some arrows between the exposures and the disease. In the COPSAC 2010, we have obtained quite many samples of these 700 children. We have vaginal samples in pregnancy. We have airway samples in early life, 1 week, 4 weeks, and 3 months of age on all children. Then every time the child is sick, they come to us. We do airway sampling as well there. We do fecal samples from 1 week to, I think the latest sample time point is 10 years. We have breast milk. We have throat swabs. We have skin samples. We have bed dust collected for the home environment. And then we characterize all these with our good collaborators at Copenhagen University. So how does the microbiome mature in very early life? This is my son 13 years ago, I guess, and my dog. And of course, you'll be exposed to microbes from the home environment. So in the cohort, we can see that the microbiome evolves very, very much, not only in the first year of life, but if you see to the left, we have the number of ASVs present. So that is the diversity, how diverse is the composition. So the number of different bacteria kind of levels out between 4 and 6 years of age. We see that the composition to the right, the composition of bacteria looks quite alike at 1 week and 1 month. 1 year separates a lot, and then kind of narrows down between 4 and 6 years. So there is this very big dynamics going on. And this is what we try to zoom in on in our studies, where we try to look at what happens in the microbiome before you actually get disease. So when you do longitudinal samples, you have some interesting possibilities. The bacteria in early life are very dynamic, very narrow in the beginning, very good at digesting milk. And then many mature species come in and take over. And we call this the microbial maturation, which is a dynamic process in the first 2 years of life mainly. And we try to measure this by having our very early samples and a sample at 12 months, so 1 year. And this is before the child is deceased. And then we looked at asthma at age 5 to try to comprehend what happened if you didn't mature sufficiently to age 1 year. So we divided that and looked at asthma risk. So does the microbial maturity at 1 year affect the risk of asthma? And what we saw was actually that if you at 1 year had a low maturity, your microbiome in the gut looked more like the early sample time points, then you were in high risk of getting asthma by age 5. Furthermore, we took this heritage that I talked about into account and tried to look at what happened if you were born to an asthmatic mother or not. And it only seemed to matter which bacteria you had. If you were born by an asthmatic mother, there the increase in risk if you had a immature microbiome was dramatic. That is the figure in the middle here. Furthermore, we tried to look at what box actually defines this immature composition. And if we look at the different top 20 different bacteria here, we can see to the right, they are the children born by non-asthmatic mothers. The orange ones are the ones who develop asthma at age 5. The green ones are the ones who don't. And in the non-asthmatic mothers, children born by those, we didn't see any difference in the bacteria at 1 year. However, in the asthmatic mothers, actually 8 out of these top 20 bacterial genera was significantly different between the ones who developed asthma and those who didn't. And what we saw was actually, I think, 7 of these were higher in the ones who didn't develop asthma, which is very interesting when we talk about possibility for modulation of this. So we could also go in and do a more targeted approach where you say which bacteria seems to be missing in later asthmatics, which are in more abundance. And by doing this, we could pinpoint, say, okay, maybe these 10 bacteria would go very nicely together in a probiotic cocktail of some kind in the future. And we're working along these lines with collaborator Søren Sørensen's group at KU to try to define this good consortium of bacteria that we hope in the future to introduce to children to protect them. Moving on to another study that we recently published is about delivery mode. So if you're born by cesarean section, you're in high risk of getting asthma. And it has been proposed that this could be through the microbiome. And we tried to explore this hypothesis here. Quite many studies have over the time done this association that C-section is a risk factor for asthma, also other immune diseases. And quite many studies have also looked at the early life microbiome, where it seems to be, especially the gut microbiome, seems to be perturbed very early and then normalizes. But no studies have looked at the two together. It's a hypothesis that they could be linked, and this was what we tried to look at in our study here. We have 700 children, 22% are born by cesarean section, and then we used, again, the same samples as before, the gut samples in the first year of life, and then looked at asthma association by age 6. So what I did was trying to define what I would call a cesarean section microbial fingerprint. So build a model that would tell me whether we could make a composition of bacteria that was the most specific on children who were born by cesarean section at each time point. So you can see to the right, I tried illustrating that with an actual fingerprint. And what we can see here is that at 1 week, the fingerprint is very strong. At 1 month, it's a little weaker, but still strong. And at 1 year, it fades out very much because most children actually normalize their composition after cesarean section by each 1 year. And then we wanted to take this fingerprint and look at what it meant for asthma. Going into how the fingerprint looks bacteria-wise, we can take the one to the left. It's a multitude of bacteria, so I think 20 bacteria goes into this model. But you can see a child born by cesarean section has less Bacteroides, less bifidobacteria, less Parabacteroides, more Veillonella, more Clostridia, more Streptococcus, Enterococcus, and so forth. That is the fingerprint at 1 week, and we did the same at the 1 month and 1 year sample time point to see what the fingerprint of cesarean section in the microbiome would look like. And then we correlated each child's composition, say how much do you resemble that fingerprint? Are you very perturbed? Or do you look very much like a cesarean section born child or not? And we could take that score and try to see whether it correlated with the later asthma. And we did that, and actually we found nothing on the 1 week. So this is where the C-section children are highly perturbed in their microbiome, but we didn't see the more perturbed the microbiome was, the higher the risk of asthma would actually get. We found similar risk of these high perturbed and totally normal vaginal-born children. At 1 month, we saw exactly the same, no increased risk of asthma here. But then in 1 year, we saw something very interesting. Even though we had this weak fingerprint, we saw that the children who carried on a cesarean section fingerprint to age 1 actually were more than double risk of having asthma over time until age 6. Then we took this observation, and then we looked at what happens between the C-section delivered and the vaginal delivered. And the vaginally delivered, there, the blue line here. So that is what you would like to look like. And then we divided the cesarean section into those who normalized, who are the orange ones, and the purple line here, who have a high cesarean section microbial score. So they still carry this fingerprint in their gut microbiome at 1 year. And we actually saw that if you normalized the orange ones, then you were quite alike the ones who are vaginally delivered. Whereas the ones who didn't normalize, they have a massive increased risk of getting asthma. And we can, of course, speculate what to do about this, because this really opens up for the possibility of saying, can we actually restore the microbiome? Can we save these children if the observation is true? Can we help them normalize their microbiome in the first year of life, so they'll avoid this higher risk of asthma? And what we saw was actually that having an older child seemed to normalize the bacteria composition. And now we're studying whether certain compositions very early on can actually aid in the later restoration process here. So with this, I would like to thank you for your attention. I would like to thank the entire team and all our good collaborators. Thank you very much for having me here.
Great. Thank you so much, Jakob. Very, very interesting talk, and also the ability to use these fantastic cohorts that you have access to. So really great. There is actually some questions coming in, but as I said, we will actually hold it till the end, and then when we do a Q&A, Jakob. And so now basically we would like to go to the final talk of this session, which is by Professor Daniel Aagaard. So I think without further ado, I think we'd like to hand over to Daniel for your presentation.
So thank you for your kind invitation. The title of my talk is What Triggers and Promotes Celiac Disease and Can We Prevent It? Lessons learned from the observational and birth cohort studies. So the incidence of chronic immune-mediated disorders, including autoimmune diseases, are increasing worldwide. The emerging increase is faster than can merely be explained by genetics. Autoimmune diseases may affect almost every organ, of which several have been associated with an imbalance of the gut microbiome, so-called dysbiosis. So during the past decades, change in the way of living from rural to urban lifestyles, diet, mode of delivery, cleaner environment, use of antibiotics, vaccination, etc., may all be potential environmental factors that have consequences for the gut microbiome. Celiac disease is one of the most frequent occurring among autoimmune diseases, with an increased prevalence and associated with changes in the microbiome composition as compared to healthy individuals. However, Most of these studies are cross-sectional with risk of reverse causality. The need of larger prospective studies are warranted in order to reveal whether or not the change in microbiome is a cause or consequence of the disease. Celiac disease affects 1 to 2% of Caucasians. It is strongly associated with HLA-DQ2 and DQ8 and cannot be developed without exposure to dietary gluten, which is the driving antigen for the disease. However, 40% of the general population carry these genes, and gluten is a universally consumed food antigen, while environmental factors have been proposed to trigger the disease. Celiac disease affects the intestinal mucosa of the small bowel, and due to an intolerance of gluten causing a chronic inflammation in lamina propria that leads to the structure of the gut epithelium. It has been hypothesized that an increased intestinal permeability of gluten antigens over the mucosal layer may activate and crosstalk with memory T cells in the immune system, which leads to a chronic intolerance to gluten. The amount of gluten, gastrointestinal infection, probably caused by viruses or bacteria, have all been promoted to promote the risk of the disease. If these promoters can prove causality for triggering disease, then prevention may be possible in genetic risk individuals for this lifelong autoimmune disorder. Prospective studies are therefore warranted in order to find candidate triggers in autoimmune disease. Since many autoimmune disorders are associated may share common underlying mechanism, despite of the candidate triggers and promoters are different. For instance, type 1 diabetes and celiac disease frequently overlap and affects approximately 10% of the patients. Therefore, many prospective studies have screened for both diseases in the general population. So this slide shows the most well-known prospective birth cohort studies performed in type 1 diabetes and celiac disease. And although the prospective design of the screenings for both diseases are shared, they differ in size when the birth cohorts were enrolled and the geographically site of origin. Many of the birth cohorts have been longitudinal followed from birth and now reach adulthood. In 2001, the National Institutes of Health decided to fund a multinational observational study on type 1 diabetes and celiac disease, the Environmental Determinants of Diabetes in the Young study called the TEDI. So what these prospective studies show in common is by prospectively collecting data and screening with a longitudinal follow-up before onset of the disease, the natural history of autoimmunity progression to celiac disease can be described. A main finding of importance from these observations is that onset of autoimmunity by seroconversion to autoantibodies to celiac disease peaks already at 2 to 3 years of age, and sometimes years before onset of clinical manifest of the disease, which occurs more often around school age. So this indicates that environmental exposures that may trigger celiac disease should be searched for before 2 years of age, and the primary prevention should focus on measures during infancy. And this offers a window of opportunity that should be done very early. So what are the candidate triggers promoters that have been found in these studies? This graph illustrates the candidate triggers that have been identified. Green arrows indicate an association with celiac disease autoimmunity or celiac disease. Red arrows, the confounders, and the gray are the still unknown and remains to be confirmed. Despite there might be differences, what triggers celiac disease in adults and children, prospective screening study points to that the genetics, gluten intake, gastrointestinal infections, and where you live are all associated and dependent on how to develop celiac disease. Moreover, there are protective effects of rotavirus vaccination and season of birth indicating a role of a gastrointestinal virus, and other factors previously associated like mode of birth, breastfeeding, time to first introduction of gluten, and the maternal diet have not been confirmed to be associated with the disease. However, important studies on the microbiome, envirome, and the metabolome remains to be performed. Those highlighted in pink are in work in progress, and results from the TEDI study are still pending as of 2021. So what is the TEDI study? The TEDI study is an observational study including 6 clinical centers located in Finland, Germany, Washington State, Colorado, and Georgia, Florida in the US. The goal of the TEDI study is to explore environmental differences in type 1 diabetes and celiac disease across diverse populations and ethnic groups in children with and without a family history of type 1 diabetes. So the TEDI study screened over 425,000 infants between 2004 and 2009 to get the children that were at the highest risk for type 1 diabetes and celiac disease. Over 8,000 children with high-risk genes for type 1 diabetes and celiac disease were prospectively followed and enrolled for a 15-year follow-up. So it has a very intense and ambitious protocol, which is common for all clinical sites. And this graph actually shows available data and sample as of January 31st last year. So as of 2020, all children have been screened for celiac disease annually from 2 years of age, which means that over 71 screening tests have been performed, of whom more than 500 children have been diagnosed with celiac disease. From this cohort, 200,000 stool samples and serum samples have been collected and almost 100,000 food questionnaires collected. So as of 2001, we now have enough cases to perform case cohort studies with credible power and prospectively collected samples prior to onset of the disease. A major finding from the TEDI study is a description of the temporal development of the gut microbiome in early childhood. This study analyzed monthly stool samples from 6 months of age using 16 sRNA sequences from 900 children between 3 and 48 months of age. What TEDI could show is that the normal gut microbiome undergoes 3 phases. First, the developing phase between the first year of life, and then the transitional phase after breastfeeding during the second year of life, and then they enter the last phase called the stable phase from 3 years and on. The covariate that was significantly associated with increased gut diversity and maturation was breastfeeding and associated with high levels of bifidobacterium. Other covariates associated with higher levels of bifidobacterium were infants being delivered vaginally, or exposed to household pets, having siblings, but also it was dependent on where you live. So the main conclusion from this study is that the gut microbiome shapes during the first 1,000 days of life, and then little changes in diversity. So if breastfeeding is associated with higher gut diversity, does it protect the children from celiac disease? Well, this slide shows the promoters that have identified so far. On the x-axis, you see to the right the higher the risk, and on the y-axis, you see the listed promoters of most importance, ranked from high to low. And what it actually shows, the #1 risk factor for celiac disease is if you have a genetic predisposition carrying DR3, followed by 1 copy of DR3. And also if you have a first-degree relative with celiac disease on the 3rd place, emphasizing the role of the genetics. Also, girls are at higher risk, and these are all well-known risk factors. But interestingly, this first paper published in 2014 in New England Journal of Medicine showed that the risk of celiac disease also differed by country, even though that you follow the same protocol in the TEDI study. And the children from Sweden were at a 2-fold increased risk as compared to the US children. So this points to the direction that there could be differences either in genetics or in exposure to environmental factors. The second most important factor was the amount of gluten intake consumed during the first 5 years of life. For every gram intake per day, the risk increased 30%. In fact, 2 grams per day at 2 years of age increased the risk of celiac disease by 3 years of age. Thirdly, a gastrointestinal infection 3 months prior to seroconversion to celiac disease autoimmunity increased the risk by 30%. So this finding was supported by an increased prevalence of enteroviral exposures in stool samples collected between 1 and 2 years of age, and increased the risk of celiac disease autoimmunity even higher with gluten intake amounts. Finally, the viral hypothesis was supported by the protective effects of a rotavirus vaccination, supporting the notion that the virus might be a trigger in celiac disease autoimmunity. But to our surprise, neither in breastfeeding were protective. In fact, it was associated with higher risk of celiac disease, and also C-section were protective. So that leads us to the prevention studies. So by identifying all these important risk factors and promoters in celiac disease from observational studies like TEDdy, we further wanted to test if celiac disease can be prevented. An interesting finding is to test whether or not probiotics have a role. In TEDdy, we observed not only differences in the prevalence of celiac disease between the countries, but also differences in the use of probiotics given to the children. Celiac prevention with probiotics is a secondary prevention trial that tested the hypothesis if 2 strains of lactobacillus, kindly provided by Probi AB here in Lund, had an immune-mediated effect on the peripheral immune response in children with ongoing celiac disease autoimmunity after 6 months of intervention. We designed a randomized, double-blind, placebo-controlled study and enrolled children with low levels of TDJ signaling ongoing celiac disease autoimmunity to either receive lactobacillus or placebo. Although the 4-year follow-up after the study ended did not show any differences in prevalence of celiac disease autoimmunity or celiac disease, the results showed quite interesting that levels of TGA, which is a marker of celiac disease autoimmunity, and NK cells decreased in the probiotic group as compared with the placebo after 6 months of intervention, as seen in the graphs to the right. So the conclusion from this study was that lactobacillus could have a dampening effect on the immune system, but longer and larger intervention studies are warranted in order to evaluate the long-term effect on celiac disease. So we extended the previous study from CIP and investigated the effects of lactobacillus on the gut microbiome by analyzing the stool samples after 3 and 6 months of intervention. Interestingly, we found that children received, or not surprisingly, lactobacillus increased their levels of lactobacillus as a sign of good compliance. But also we found abundances of Akkermansia and Dialistin, as shown in the graph to the right. Akkermansia is a mucin-degrading bacteria that feeds on the host mucin layer. Akkermansia mucinophilia has been found inversely associated with celiac disease and chronic inflammatory disorders. So it seems that if lactobacillus happens to restore Akkermansia to normal levels in children with celiac disease immunity and develops a healthier gut flora with the mucin level. However, the interaction between lactobacillus and Akkermansia and its long-term effects on the celiac disease remains to be investigated. So in 2018, we started the next generation of screening infants on the ongoing ASTRID study. The goal is to screen 55,000 newborns for the HLA risk genotype for diabetes and celiac disease. Those children that carry the highest risk gene for celiac disease, namely the DR3 homozygotes, which comprise about 1.8% of the general population, will be invited to a randomized clinical trial of either receiving a gluten-free diet or lactobacillus or placebo or as a normal diet for the first 3 years of life, and then will be screened for celiac disease until 7 years of age. So based on the TEDdy study, we expect actually 20% to develop celiac disease on a normal diet. The goal is to enroll 600 children and 200 in each arm. And the study has enrolled participants since August 2018 and will continue to enroll children to 2023. To date, 14,000 children have been genotyped, of whom 250 have been eligible for the PRECISE study, and we have actively 8 children in the study. So we plan to follow up the study until 2030. So to summarize my presentation is that dysbiosis is associated with several autoimmune diseases, including celiac disease, but we need larger prospective studies to test for true associations. So based on the observations from the prospective studies like TEDDY, we can show that the gut microbiome develops during the first 3 years of life. Onset of celiac disease peaks before 3 years of life. Genetics, HLA, are the strongest determinant of celiac disease, but there are other candidate promoters such as high gluten intake and gastrointestinal infections, possibly by an enterovirus that may trigger the disease. Clinical trials with probiotics, like lactobacillus, show dampening effects on the peripheral immune response and positive changes in the bacterial flora in children with celiac disease on immunity. So based on these findings, we have now initiated new prevention studies like PRECISE to test the hypothesis if early gluten-free diet intervention or probiotics given to children at the highest genetic risk will prevent them from developing celiac disease. So finally, I'm not alone on this to perform these studies. I would like to send my special thanks to Karin Andrén Aronsson, Elin Oskarsson, Åsa Håkansson, Jörn Molin, Charlotte Brundin, Åsa Wimar, Probi AB, and TED Group. Thank you so much for your attention.
Thank you, Daniel. Thank you for the great talk. And now we're actually going to be doing the Q&A session. So with that, we'd like to start looking at some of the questions. So please write in if you have anything you'd like to ask any of the speakers. And we're here and we're ready. And I'll just start to go through some of the questions. And actually, one of the first questions I think will come to you, Jakob, from Karen Broholm, who says, Dear Jakob, do you also collect blood and urine samples from the mothers during pregnancies with these fantastic cohorts you're working with?
Yes, so we have blood during pregnancy. We don't have urine on the mothers, but blood we do have. We haven't used the blood exactly to study microbiome interactions, but that is definitely something that we can look into in the future. I don't know whether you had some specific questions regarding the blood and urine or what you wanted to compare. We have done metabolomics analysis on the blood, but not related to maternal microbiome yet.
Yeah. I think the question was more just probably about collecting these samples. It's such a fantastic cohort you have available.
So we have collected everything. These it's so fantastic with these families. They come to us. We have actually, I think, a 92% 6 years follow-up. So they keep on coming, even though we actually, to some extent, torture the children a little bit.
You torture the children?
We collect everything we possibly can. But
it's fantastic. Fantastic research. Great, thank you. I hope that answers the question. So we have now a question for Asa. So the question is, Dear Professor Asa, are there any published observations or work in progress that the macrophages may have a specific positive or adverse effect in relation to the indication like sarcopenia in the elderly?
Thank you so much. Not that I'm aware of, at least, not from our group. Not so far, but it's an interesting area.
Okay, thank you very much for that. So we have several other questions coming in. An anonymous question here. Jakob, this question comes to you again. So it says, thank you for a great presentation. It was. Few studies have established that fetus also have a microbiome. Have you been doing any analysis of sort of fetus microbiome, or could you give us more insight if that actually exists, and will there be any significant differences?
That is still debated in the community, and if you ask different researchers, you will definitely have different opinions on that. If we have a fetal microbiome, it will be very low biomass, and it can be to some extent explained when you see how much a cesarean section delivery can actually change the early life microbiome. Because if you had already a very established microbiome when you come out, then you wouldn't have this massive perturbation of just this one procedure. So I do believe that there may be bacterial remnants there, but probably in very, very low amount.
Okay, yep, great. And it's actually, I was just seeing, I'm scrolling through here, I actually just saw an interesting link, and I'm just trying to capture it. I think Sarah's moving the questions around for me just to challenge me. Modern technology. So Jakob, it's actually linking to this. So can you speak to the difference between a voluntary cesarean section and medically induced cesarean sections, and how that is actually affecting the microbiome? And that's something I think is very interesting.
Yeah, so we have tried studying that also on the population base. So Denmark is so great because we know everything about everyone here. We can link to the CPR number and actually do great research in the public registries. And there we actually tried linking first to the asthma risk. And we actually saw that if you come out by a scheduled or planned cesarean section, then you're actually in even higher risk than if you're born by an acute cesarean section, where the water has to some extent broken and maybe a little bit of bacteria has occurred to the child. Regarding the microbiome, so I also looked at it in this work where we looked at the cesarean section. We did look at different types. We don't see very strong differences in the early life microbiome according to delivery type, but other studies have actually shown some differences there. A little bit on that. Of course, you can also be vaginally delivered and have antibiotics during birth. So about 10% of women will probably have antibiotics there. And there we can actually see a very interesting thing, that if you're born vaginally, but mother is treated, then your microbiome will develop in the middle of vaginal without and cesarean section delivery across the first year of life.
Okay, that's very interesting. And I think I'd like, if possible, to actually extend that question to Daniel and actually ask, do we actually have any factors there that you could consider from birth or cesarean sections antibiotic use that actually would be prevalent or increase risks or associate with celiac diseases from your research?
Well, what the prospective studies show is that neither C-section or use of antibiotics. Some studies show that the use of antibiotics actually may trigger celiac disease, but others, like TED, did not show that association. So we don't know yet. I mean, these are observations. I think we need to go to the microbiome and do prospective studies and see if the changes is associated with celiac disease later in life.
Great. Thank you, Daniel. I'd actually like to stay with Daniel because we just had a question come in. And the question would be, is from Mikhail. Will you be undertaking more studies related to environmental impacts or risk of developing autoimmune diseases in the general, or more specific to celiac disease?
That's a good question, because these observational studies are actually selected from the general population. In fact, we are expanding our observations also on thyroiditis, autoimmune thyroiditis in TEDdy. But then again, these are selected also based on their genetic propensity for developing either celiac disease, type 1 diabetes, and thyroiditis. So it means that if you're going to look for other studies, you may have either other selection criteria. So I think that's what we can do at the moment.
Fantastic. Yeah, I think it's great. And the ability to be able to use these cohorts, as we've been discussing in the areas. A question, actually, I'm jumping back to you, Jakob. A question from Natasha. Thank you again for a fantastic presentation. It says, such a rich data set. Now, this is a slightly different. Have you looked at the neurological development factors or data when you associate with the microbiome alterations?
The quick question is no. So we have extremely strong neurological data, actually ongoing to 10 years, where we have a great psychiatric evaluation of the same children here. We have neurodevelopment of both gross motor and language and cognitive development, but we haven't looked at it yet regarding the microbiome, and of course that will be the next steps to do in our cohort.
Okay, great. Thank you. And a question here from Henrik. It says, again, thank you, Jakob, for a great talk. And I think this is a general question. We can actually touch on this maybe to several of the speakers today. What is the most important with respect to immune development in early life? Is it the composition or activity of the infant microbiota? Any thoughts on this? And I think I would like to sort of maybe touch on that to all three of you, actually. So, Jakob, why don't you go first?
That's a difficult question, I guess. So the quick question is, we don't know. We try to evaluate different diseases in different organ systems. So we also look at the airway microbiome early life, and we also see associations there between the early life microbiome and asthma development. For the gut, we believe it's a matter of immune training, but we haven't got a very good tool to actually measure this immune training over time. So we try to of course look at when does the disease appear. So the composition I talked about with the maturation, that's actually also associated with allergies. So if you have this immature microbiome, we will see that the allergic asthma phenotypes are actually more pronounced, which probably suggests that it's something with longer-term immune programming that you actually learn from your microbes which to react on and which not to. Yeah, but maybe some of the other speakers can
Exactly. I think
can help me here.
Yeah, ask the same question to Asa and then Daniel also. Asa first.
Yes, should I start? Well, I'm sure that it's both the composition of the microbiota and also the quantities of different genus and species, and also maybe on strain level that will affect, but also the activities of the microorganisms that will affect the immune system development in children. So, but it's a lot of research is necessary to be able to draw any conclusions about it. It's very complex. The interactions are very, very complex, and it's sometimes hard to discriminate between different species and different genus and what the effect they actually do have on different phenotypes of the immune system.
Great. And Daniel?
Well, I would say I actually don't know. I think that many of these studies that has been performed, we don't know if it's a risk causality. I mean, what does the microbiome do? Does it, the disease cause a change in the microbiome, vice versa? I think that we need larger prospective studies, collected information prior to onset of diagnosis in order to link the changes in the microbiome to maybe SNPs, maybe to other genetic factors and the diet, and also the infections, at least when it comes to celiac disease. We have one, as I said in my talk, we have one of those studies actually ongoing, which will be a huge effort to try to understand how the virome and microbiome affects the risk or associated with the risk of celiac disease later in life. We will collect, I think, almost 20,000 samples from birth to onset of disease, and then try to link that to genetic factors and other environmental exposure to see how that affects the risk.
Great.
But the study is going.
Okay, thank you, Daniel. I have a quick question for you, Daniel, and the final one for Asa. Actually, Daniel, it says, you looking at the effects of lactobacillus, and this is close to my heart, of course. How about looking at other probiotic bacteria like bifidobacterium? Daniel.
Yes, that's an excellent suggestion. And in fact, there are studies that are looking into that, and there are clinical trials that just recently started that will look on the effect of bifidobacterium on the risk of type 1 diabetes and celiac disease, but they've just started. So we have to see and wait and if that has an effect, but it's a great suggestion. Bifidobacterium should definitely look more into.
Super, thank you. And I think a final question to Professor Asa. Do you have any plans to conduct research on maternal nutrition health pre-conception and its potential correlation to the fetus-child microbiome? So Asa, final question.
Sorry again, can you please repeat that question?
I'm sorry, I said it a bit fast.
Sorry.
Do you have any plans to conduct research on maternal nutrition or health in pre-conception and its potential correlation to the fetus or child profile of the microbiome?
Not at the moment, actually, but again, it's very, very interesting, and I would like to do that. I think it's important to also look in the mother and see how it's affect the child after delivery and so on. So I absolutely think that it should be done. Right. But for now, we don't have any plans for doing that.
Great, and thank you. And I think there are more questions coming in, and I think these, of course, could be answered at a later stage. But I would just like to thank the three speakers to say thank you for some great insights to the research you're doing. I think it's fantastic, the capabilities that we have in the local region in Medicon Valley, showing the learnings we can do and the fantastic research to actually start to understand immune functions and immune development and the links with the microbiome.